we can go ahead and get started. My name is Hillary Peterson. I'm the president and webmaster of Maine Entomological Society. Thank you so much for taking the time to be here this evening. This is our third webinar for the, 20 to 21 winter season. Oh, can everybody hear me? Okay. So this webinar idea was sort of cooked up at the annual meeting back in October. We were trying to think of ways that we could sort of have a little more interaction over the sort of off season. Maine Entomological Society does have lots of field trips in the spring and summer. You can find out about those at our website, mainentosociety.org. So without further ado, I'd love to introduce Patty Jones, who's an assistant professor of biology at Bowdoin College and also the director of the Bowdoin Scientific Station on Kent Island. She's originally from Cambridge, Massachusetts. She has her biology and history of art from Cornell University and a PhD in integrative biology from the University of Texas at Austin. She returned to Cornell as a postdoctoral researcher for 3 years before joining the Bowdoin faculty in 2017. Patty has worked with a range of different systems, including lobsters, butterflies, bats, and most recently, bumblebees and seabirds on Kent Island. She lives on the Androscoggin River in Brunswick, where she rows and has 2 little boys. Thank you so much, Patty.
Thank you for introducing me, Hillary. I'm coming with the caveat of, as you can tell, to one very little and a 2-year-old who's going to bed right now, and a dog who's whining about his ball he's lost under the sofa. So the normal chaos of talking from home in this era. And I hope that the disruptions will be minimal. We'll do our best. Shall I just go ahead? You can the screen sharing looks okay, Hillary, and everything? Okay, great. I'm going to talk to you about my research that I've been doing, predominantly research that comes from my lab at Bowdoin, but also some that we've done on the field station on Kent Island, which I'll introduce a little bit for those of you who aren't familiar with Kent Island. But I'm really going to be talking today about the enigma that is the plant toxins present in flower nectar. And as many of you, I'm sure, know, lots of plants have toxic compounds in their leaves that defend their leaves from herbivores. But many of those plants, including this milkweed that you see here, also have those same toxic compounds in their nectar. And this has long mystified ecologists. Why would, when you think about nectar as existing as a sugary reward for visiting pollinators, why would a plant have a toxic compound in their nectar? You'd think that would deter the pollinators or potentially poison them. And a number of hypotheses have been proposed for the presence of these toxic compounds in nectar. One is that perhaps these toxic compounds are present in nectar simply because they're present in leaves. They make leaves distasteful and toxic to herbivores and defend the leaves of plants, and that's necessary for lots of plants or to the advantage of lots of plants. But there have also been a number of studies that have shown that the compounds that are present in the nectar are both quantitatively and qualitatively different from the compounds that you find in leaves. And that indicates that perhaps there actually are selective pressures that are driving the plants to have these toxic compounds in their nectar. So what are some of the possible explanations for why plants would have toxic compounds in nectar? One of them is that perhaps they filter for more effective flower visitors. So perhaps something like ants that will often rob nectar from flowers, damage the flowers, not pollinate, but consume the nectar. Perhaps they might be deterred by a nectar toxin, whereas something like a visiting bee wouldn't be deterred. So perhaps the presence of those toxins actually selects against particular insects, mostly insects, that are not very good, not very effective pollinators. Another potential hypothesis is that maybe they actually are there to deter ovipositing herbivores. So lots of insects, as you know, have nectar-feeding adult stages and herbivorous larval stages. So we think about lepidopterans that perhaps are actually visiting flowers as adults and using cues from nectar as signals about where to oviposit. And maybe the presence of those toxins in flower nectar really functions to influence the decisions being made by ovipositing insects rather than necessarily by pollinators themselves. Another potential explanation is maybe these compounds change pollinator behavior, so they have some kind of pharmacological, psychological effect on the insect that improves its pollination behavior. That's what I'm going to talk about quite a bit more today. And then a final explanation, which actually it seems like in a lot of systems may be the best supported, is that maybe these compounds prevent microbial degradation of the flower nectar. So these compounds, a difference between a poison and a medicine is the dosage, and they often at low concentrations can have very good antimicrobial effects. And if you think about honey is very quite antibacterial and antifungal in a lot of ways, and that's mostly due to some aspects of bee saliva, but also a lot of it has to do with compounds that are present from the plants, acquired from the plant flower nectar. So in my research in the lab and in the field, I investigate some of these different hypotheses using a system of bumblebee colonies connected to flight arenas in the lab and wild caught field bumblebees on Kent Island. And I'm going to talk to you today also about some of my research coming out of my postdoc, which was looking at some of these questions with herbivores as well, and with monarch butterflies in particular. So the first section of the talk, I'm going to focus on these first three hypotheses for the explanations for the presence of toxic compounds in nectar, using both bumblebees in the lab as well as monarch butterflies. And then the next section of the talk, I'm going to be talking about this hypothesis that perhaps these compounds change pollinator behavior, and particularly looking at caffeine, which we know is present in citrus and coffee flower nectar, and trying to understand how the presence of caffeine in flower nectar can influence bee behavior in a way that actually might benefit plants. And then in the final section, I'm going to talk about a consequence of microbial degradation, which could be ethanol production or a buildup of ethanol in flower nectar, and how does ethanol, that alcohol, how does the presence of alcohol, the fermentation of flower nectar, influence bee behavior in a way that might be negative for the plant? So to start with this first section, I'm going to talk mostly here about milkweeds. Milkweeds are a great system to be able to ask some of these questions because they have, they're reliant on a number of pollinators. Largely, we know that milkweeds are pollinated by large-bodied Hymenoptera. So bumblebees, wasps, carpenter bees, big-bodied bees in particular that can get their legs down into the correct parts of the flowers. Milkweeds are a very diverse group of plants. They have quite a bit of diversity in flower morphology, leaf morphology, and toxins. Milkweeds are quite toxic. They contain what are called cardenolides or cardiac glycosides. The one you might be the most familiar with doesn't come from a milkweed. It actually comes from foxglove. This is digitoxin, which is commonly used as a heart medication. And milkweeds actually were used by Indigenous Americans for congestive heart failure because it had that same kind of effect. These cardiac glycosides have that effect. And as a sort of interesting side note about cardenolides in general, and this one from digitoxin that comes from foxglove. So if you overdose on cardenolides, if you consume too much of them, it's been shown to have the effect of a yellowing of your vision and yellow halos in particular around objects. And we know that Vincent van Gogh was prescribed extracts of foxglove to try to treat his seizures. And it has been proposed by some doctors that some of Vincent van Gogh's vision in his art was actually perhaps influenced by an overdose on these cardiac glycosides that he was being prescribed from foxglove. Do that yellow halos, if you think about Starry Night or sunflowers, may actually have been somewhat influenced by, because that's the visual effects that overdosing on these cardenolides have. So cardenolides or cardiac glycosides are found in a number of different plants, but are ubiquitous in milkweeds. And milkweeds in particular actually contain a whole range of them. So any common Asclepias syriaca, the common milkweed you see on the roadsides here, may contain upwards of 15 different cardiac glycosides in their different tissues. And they have different concentrations of these compounds in different tissues. And corresponding with that, they have a whole suite of specialist insects that consume different tissues of milkweeds. And each of these insects has an adaptation, with the exception of the bumblebee in the middle here. Each of these insects we know has an adaptation that allows them to not be negatively affected by these cardiac glycosides. So they're resistant to the toxic effects of these compounds in particular. But bees, on the other hand, we know these compounds occur in nectar, and we don't know much about how they impact the pollinators that we know are important for milkweeds. And large-bodied Hymenoptera like bumblebees are particularly important pollinators for milkweed, which has a really interesting pollination system. So like orchids, their pollen is not a loose dust, rather it's locked into these wishbone-shaped structures, which are called pollinia. So bees foraging from milkweed, you'll actually see, if you watch them, you'll see these, they have these little yellow, there is an arrow to it here on this bee's legs, but these little yellow wishbone-shaped structures, which are the pollinia of the milkweed, stuck to their legs. So they can't collect pollen from milkweed, they can only collect nectar, but the milkweeds are quite dependent on them getting their legs down in the right positions on the flower to extract those pollinia. And we know from previous research that milkweed nectar contains these cardenolides, these cardiac glycosides. So this is from a paper looking at a range of different milkweed species, all in the same clade here of milkweeds, and looking at the concentration of their cardenolides in their tissues. And interestingly, different species of milkweed contain different levels, different concentrations of these cardenolides. And we know that they have a lot of variation in how much of these cardenolides they have in their leaves. And so it's not surprising that they also vary in how much they have in nectar. But importantly, there isn't a fully corresponding match. So some of the species that have really high nectar concentrations aren't necessarily the same species that have high leaf concentrations. So that indicates that perhaps there's actually an adaptive explanation for why these compounds are present in nectar. And it's not just that they're circulating levels for leaf defense that are therefore spilling over into flower nectar, for example. So one of the things that we're working on in the lab is looking at how how these presence of these compounds in nectar influences bee consumption of nectar. So this is really preliminary data where we're sort of we're providing bees with different concentrations of, this is a commercially available cardenolide, which is called wabane. And all I sort of wanted to remind myself to show you here is that as the concentration of these cardenolides increases, bees are consuming less of them in nectar from our early preliminary data. It looks like this blue line here is just a sucrose solution. And over time, bees are still continuing to consume quite a bit of sucrose solution, but our bees who were fed nectar with cardenolides either died or they consumed a lot less of that solution than they do just a straight sucrose solution. So the first thing that I wanted to do was to understand, how does the presence of these compounds in nectar affect the way that bees behave? And how does it affect whether or not they visit flowers? So to do that, I use these commercially available cardiac glycosides. And what I do is, this is very sort of representative of the type of bee behavioral research I do in the lab, where I train bees, I present bees with artificial flowers that are blue. In this case, for this experiment, were blue paper discs. I often use little square chips of color, which I'll show you, which allow me to clean them, remove any scent marks, and just control. I've got color and I'm manipulating nectar chemistry. So what I've done in this experiment was I gave bees one flower color with a nectar solution with cardenolides added to it, and one color with just a nectar solution. And then those bees were presented sequentially with those two options. And then I allowed them to forage in an arena with a combination of those two choices. And I looked at, where do the bees choose to visit? So how much I presented them with this, a color that has been paired with just a straight sucrose solution, just sugar solution, and one with this toxin. Do they avoid those flowers that they had learned were paired with that toxin? And what I found was that when I looked at individual bee visits, so here I'm looking at just sugar solution versus sugar solution with these toxins added, and I look at the number of visits made by individual bees, I don't see a difference. So it doesn't seem like the individual bees actually avoid the presence of these compounds in flower nectar. And this is at the highest natural concentration that we know at which they occur in milkweeds. When I did do it at 10 times that concentration, so at unnaturally high concentrations, they do avoid them. So they can either taste them or they have some kind of a deterrent effect, but at high natural concentrations, we don't see any deterrence. But then I thought, okay, well, this is just one foraging trip. This is one bee making one set of choices. What if we let a whole colony of bees forage on these compounds for a long period of time? So to do that, I take these are commercially available colonies of bombus impatiens that I work with in the lab. And I have a whole colony of impatiens, and I connect them to my foraging arena, and I allow them to forage from these two different options, these two different colors of flowers, one with toxins and one without. And I look at, over time, I look at what are the numbers of flowers that those bees visit of those two options. And in this case, I found that over the course of 5 days of the whole colony foraging on these flowers, I do see fewer visits to this high natural concentration of cardinalides and nectar. So when a colony of bees is allowed to forage for a long period of time, they do seem to begin to avoid the presence of these milkweed toxins. So that makes some sense when you think about the ecology from the plant's perspective, that you wouldn't expect plants in the field to have concentrations as high enough that they would be really deterrent to their pollinators. But bees in the field are, as you all know, are foraging on a wide range of things, especially bombus impatiens is very generalist. And so they're probably not accumulating a ton of these toxins in their colony over time. But if I do force them to only consume milkweed nectar, this toxic milkweed nectar, they start to avoid it over time. But then I was interested in this sort of second adaptive hypothesis that I mentioned at the beginning, which is perhaps the presence of these compounds, really what they really do is they deter ovipositing insects. And the ovipositor that we know is particularly important for milkweeds is the monarch butterfly. So monarchs consume milkweeds as larvae. They then sequester those toxins into their own tissues, which have emetic effects on their bird predators, make their birds who eat them, such as this blue jay, throw them up, or who sample them, throw them up. And I did quite a bit of research on the chemistry of sequestration of these toxins by monarchs. So this is looking at 4 different milkweed species. And I'm looking in these gray bars here, I'm showing you the concentration of the toxins in the leaves of the plants. And then the white bars are the concentrations of the toxins in the wings of butterflies who were reared on those plants. And so monarchs are really greatly influenced in terms of how much toxin they're able to sequester by how much is available in the leaves of the plants that they're eating. So that a monarch who's reared on a high cardinalide milkweed may have 4 times, maybe sort of 4 times as toxic, have 4 times the concentration of toxins as a monarch reared on a low cardinalide milkweed species. So we know that the cardinalide concentration is very important to the defense of the offspring that are reared on that plant. And as I mentioned, there's some correlation between concentrations in flower nectar and concentration in leaves, but at the same time, they're not completely corresponding. So there are significant differences, both qualitatively and quantitatively, in the concentrations of cardinalides and the types of cardinalides that occur in nectar versus ones that actually occur in leaves. We know from research with other species, so this is Manduca, the hawk moth, and we know that, and Jimson weed, I think it is, we know that these hawk moths are influenced by the presence of toxic compounds in the nectar of this Jimson weed in terms of when they're making oviposition choices. So this is not a totally novel concept of lepidopterans being influenced by nectar chemistry when they're making egg-laying decisions. So I wanted to look at this in monarchs, and what I did was I trained a group of monarchs in a flight cage. I gave them an array of artificial flowers to feed from, one color of which was paired with cardinalides and nectar, and one with just straight sugar solution. I allowed them 5 days. They're happily mating and free flying around, foraging only from these flowers that I've offered them. And then I took the mated females from that group, and I presented them with a set of milkweed plants paired with those flowers. So this is what that sort of looks like. And in this case, I've collected these milkweed plants were reared from seeds that all came from the same seed pod, which is really a convenient thing that you can do because it means that the plants are full siblings, basically. They're very closely related and very similar, with the exception of the fact that I've manipulated this artificial flower that I've added to these plants to look at how that affects how that nectar chemistry that I've added might be affecting oviposition of the monarchs. And then I looked at the number of eggs laid by these female monarchs on these 2 different plant options paired with different artificial flowers with different nectar chemistry. And what I found was that monarchs laid fewer eggs on plants that have been paired with this high cardinalide nectar. So this nectar that's a high natural concentration, the monarch females laid fewer eggs on those plants. So that indicated that maybe in milkweeds, one of the reasons that these cardinalides are present in nectar is that it actually might function to deter ovipositing insects. But there's probably a threshold at which they're sort of contained at a low level by the fact that at really high levels, they deter pollinators, but there's selection on the plant to elevate those levels in nectar in order to deter these ovipositing insects, which is kind of an interesting trade-off from the plant's perspective. And I'm continuing to work with milkweed in the lab quite a bit. I'm interested in other factors using, obviously the things I've showed you are super artificial flowers, which have lots of advantages. I can control things, all the different factors except for nectar chemistry. But milkweed flowers, these, for example, are two different flower morphs of the tropical milkweed, which you often see as an ornamental around, Asclepias curassavica, which can be either orange and yellow or all yellow. And when we looked at the nectar chemistry of these different flower morphs, we found that some of the all yellow morphs had much higher concentrations of nectar cardinalides than the red morphs did, any of the red morphs. And so we're continuing to do this work with some real flowers in the lab and looking at how bees are making choices between these flowers that differ in their nectar chemistry as well as differing in their flower color, and how those different factors of color and chemistry might actually be interacting to influence bee choices and bee behavior. I'm also collaborating with my postdoc advisor, Anurag Agarwal, who's at Cornell, on a grant looking at the concentrations of these cardinalides in different plant tissues, so in roots and in leaves and in phloem and in seeds that are all consumed by different insects, and understanding the trade-offs from the plant perspective of the distribution of compounds to those different plant tissues and how resistant different insects are and how specialized they might be to be immune to the effects, let's say, resistant to the effects of the particular compounds that occur in leaves versus the ones that occur in roots. So the distribution of chemistry across plant parts. So that was this first section talking about nectar chemistry and how nectar chemistry is influenced by the presence of how insect behavior is influenced by the presence of this different nectar chemistry. And for the second section, I want to talk to you about some pharmacological effects of nectar chemistry, which is sort of a different aspect of my research. And particularly a compound that I've been very interested in working with recently has been caffeine. So we know from previous research that the presence of caffeine in flower nectar, and caffeine occurs in coffee flower nectar, which is maybe not surprising, but also somewhat surprisingly in low concentrations in citrus flower nectar. We know from previous research from the UK that the presence of caffeine in flower nectar enhances bee memories for floral odors. And this is research done with honeybees with this PER, proboscis extension response research, where you've got a tethered honeybee and you present them with different floral odors and you look at their ability to learn and remember those floral odors. And this research showed that bees remember floral odors better when it's paired with caffeinated nectar than with its just straight sucrose solution nectar. So I was really curious about, okay, they remember odors better, but how does it affect their behavior in more of my free-flying assays that I do with these bumblebees? And there's a particular behavioral metric that I was interested in, which is what's called floral constancy. And if you're not familiar with floral constancy, this is a property of bees to, once they visit one particular, and lots of other insects actually, flower visiting insects, when they visit one particular type of flower, they'll continue to visit that type of flower, skipping over rewarding alternatives in order to stay constant to a particular flower type. And this was noticed in the field by Aristotle and by Darwin, observing honeybees in their gardens. But it's also been shown for a wide range of other insects, and it's been proposed that it makes the foraging bee more efficient because they only have to learn to handle one particular flower type in a foraging bout, let's say, and then they can more efficiently extract resources by focusing on that flower type. I'm particularly interested in floral constancy because it's this nice intersection of learning on the bee side of learning a particular flower type and then sticking with it. And that really benefits the plant because that means that that bee is only carrying conspecific pollen or mostly carrying conspecific pollen. Whereas if they were visiting a wide range of plant species, they'd have a mixture of heterospecific pollen, a range of different species of plant pollen on their bodies. But if they're only visiting one flower type, they only have that species pollen, which should enhance plant pollination. This is one of the things that we worked on at the Bowdoin Scientific Station on Kent Island. So Kent Island, Bowdoin acquired in the 1930s. It is just south of Grand Manan, right at the sort of Maine Canadian border. It was originally established as an ornithological conservation area and ornithological research station. We still have really amazing long-term data sets on Leach's storm petrels going back to the 1950s, savanna sparrows and tree swallows going back to the 80s. That we continue to do research on with collaborators from the US and from Canada, from a range of institutions. We also work on marine intertidal ecology questions, and increasingly recently on insects and on bees in particular. And one of the things, Cat Island is a wonderful place to work with pollination questions because it's a fairly limited and contained pollinator community. So we have no introduced honeybees. For example, the flowering plants that are there are a manageable number because it's a small boreal island to learn to identify for students. And but we have a number of bumblebee species. We mostly have Bombus vagans, but we have a few other species there as well. And it's a great opportunity for students to do things like in the field. This is a student, Danielle Horn, who's recently graduated from Bowdoin. And what she did was she looked at the floral constancy in the field of Bombus vagans. So this is the mean number of flowers, individual flowers that a half-black bumblebee would visit before they switch to a different alternative. And she showed that these bees are more constant to some flower species than others. So for example, they're more constant to yellow rattle than they are to dewberry flowers. And one of the things that I'm particularly interested in is what are those factors that are driving that increased constancy? Is it higher sugar solution? Is it higher volumes of sucrose rewards, of nectar rewards? Is it higher concentrations of nectar rewards? Is it color? Is it chemistry? What are some of those different factors that are influencing constancy, which might influence both the bees learning and also would have effects for the plant's pollination? Other research from James Thompson's lab has shown that caffeine in floral nectar enhances the deposition of pollen by bees. So he had this brilliant experimental design using artificial pollen, sort of a fluorescent powder, and using these little traps that the bees would pass under and get this artificial fluorescent powder stuck to them. And then when they'd visit another little artificial flower, they'd deposit that pollen. It would get stuck to some sticky paper. And he showed that at the concentration of caffeine that's present in citrus flowers, there's more deposition of this artificial pollen on those flowers than there is at either with no caffeine or at coffee flower levels of concentrations of caffeine. So it seems like these intermediate concentrations of caffeine are somehow enhancing deposition of this artificial pollen, at least, which is probably a pretty good metric for pollination. But I was really interested in, okay, there are different behaviors that bees could take that would do that. So is it that they're making more visits to those flowers? Is it that they're spending longer there? Is it that they're grooming there? Something different about their behavior that's depositing more of that pollen? What is it about the way that they're behaving that's increasing this deposition of pollen? So to look at that question, what I do in the lab is I take my colony of bumblebees and I attach them to a foraging arena where I present them with a field of artificial flowers with either caffeinated nectar or not caffeinated nectar. And I allow those bees to forage for an extended period of time from those particular flower types. And then I provide that bee with an option where there's the flower that they were trained to, and then I've introduced a novel flower type. So here they were trained to the blue, and I've introduced a novel white, and I'm looking at how constant are they to that blue that they have experience with versus how much are they willing to switch and investigate this novel flower, this white flower that I've introduced, and how does that chemistry affect that switching to a new flower type. And what I found with caffeine is that, interestingly, caffeine actually seemed to decrease in my experiments the proportion of visits that bees were making to the color they were trained to. So it seems to be decreasing constancy, and this is at this citrus flower concentration. So they're actually making fewer visits to the color they were trained to and more visits to these novel, let's say white in my example I gave you, flower that I've introduced. But I was curious then about, okay, well, in Thompson's experiment, he showed that they're depositing more pollen. What other ways might they be differing in their behavior that could result in more deposition of pollen? And what I found is that, what I did see is that almost on on those trained flower colors that the bees were going to, when the nectar was caffeinated, they were spending longer time. So they were spending more time on those flowers when it had caffeinated nectar. than bees that were not foraging on caffeinated nectar. They were also spending longer in flight, so they were sort of having longer visits in total. And one possibility is that the higher deposition of artificial pollen is driven by longer time that bees are spending on flowers more than it is necessarily by numbers of visits that they're making, which is kind of an interesting effect that we're seeing. And we're continuing to work on this with a range of other experiments, trying to explore this further using caffeine in particular. My personal interest, I worked for my PhD on bats and on social behavior in bats. I'm really interested in social learning in bees, and bumblebees don't do the waggle dance of honeybees to communicate social information, but they will join other bees at flowers. And they're more likely to join other bees at flowers under particular circumstances. So when there's a risk of predation, if they have experienced something, a simulated crab spider predator, they then are more likely to visit flowers where they see another bee present. If they don't have any information, if they're foraging from a completely novel set of flowers, they're more likely to go to flowers where other bees are present. So we know that they're influenced by the presence of other bees on flowers. And I was interested in whether chemistry could be one of the factors that is affecting when bees join other bees at flowers in particular. So to do that experiment, to ask that question, I again trained bees to a set of artificial flowers with either with just sucrose or with caffeinated sucrose. And then I presented an individual bee with an array where half of the flowers had dried pinned demonstrator bees added to them. And we know there have been a lot of experiments, mostly with Bombus terrestris in the UK, but looking at bees, bumblebees are quite responsive to oven-dried, pinned conspecifics. And they respond to those very similarly to how they do live conspecifics. Not quite as strongly, but quite similarly. And it's a nice controlled way to be able to look at this joining behavior. And what I found was that when flowers were caffeinated, bees actually made less joining visits. So they were sort of less social in some ways when they're foraging on this caffeinated nectar than when they're foraging on less caffeinated nectar or non-caffeinated nectar, which I'm particularly interested in how these chemical compounds are influencing this social behavior as well as their foraging behavior in general. So I'm pretty excited about this result. And we're pulling it apart with some experiments with live bee demonstrators as well, currently in the lab. So this final section that I want to talk to you about is about alcohol. So as I mentioned, one of the main hypotheses for why leading hypotheses, I should say, perhaps, for why plants contain these toxic compounds is that they have antimicrobial and antifungal effects. And therefore, I think that sort of the next set of logic will be, well, there must be some negative consequences of having microbes or fungus in your nectar for the plant. And perhaps it is fermentation of that nectar and an accumulation of ethanol, particularly in flower nectar. We know that yeasts occur in lots of flower nectar, and yeasts that make Saccharomyces yeast that make that do fermentation and make ethanol. And we know from other research that flower nectar, for example, this palm, can have nectar concentrations of ethanol as high as 3.5% or so. And there's actually a specialist shrew that consumes only the nectar of this palm and has a altered alcohol dehydrogenase enzyme in order to be able to cope with the fact that all they're doing is drinking 3% beer all the time and consuming nothing else. So nectar fermentation is quite common, we think, in the fields, and likely having consequences for animals that are consuming that nectar. So to do this research, what I did something slightly different, whereas I connected my colony of bumblebees to an array of two different options, one with just sucrose and one with sucrose with ethanol added at increasingly high concentrations over the course of multiple days. And I found that bumblebees started to avoid the presence of this ethanol in flower nectar at pretty low concentrations. So at about 2%, we started to have, by volume, we started to have fewer visits to the flowers that have ethanol added to them than just sucrose flowers. And that difference became more dramatic at very high concentrations. Amazingly, bees will still consume 20% volume to volume ethanol in nectar, but they've certainly avoided at that high concentration. I was also curious about flower time and found that the presence of ethanol in nectar didn't affect the amount of time that bees spent visiting flowers until I got to very high concentrations of about 20%. Did bees actually spend shorter time visiting flowers that had that very high ethanol concentration? But as I mentioned, I'm really interested in this joining behavior. And so what I also wanted to do was look at how the effect of this alcohol in nectar affects bee joining. And to do that, this is all from videotaped trials. And so I could look at live bee joining behavior. What do bees actually do when they can join each other at flowers? And I found that when bees join each other at flowers, so these darker bars are the flower visit times when the bee has joined another bee versus the lighter bars are when a bee has visited a flower alone. And at these higher ethanol concentrations, though interestingly at not all of them, and this is a frequent thing I have with working with ethanol, where I have really inconsistent results across concentrations, but at some of these ethanol concentrations, bees are spending much longer at flowers when they join other bees than when they visit flowers alone, which is the opposite of what we found with caffeine. And so this is kind of exciting that these compounds could be having effects not just on the foraging behavior and choices of bees, but also on how that interacts with their social behavior. So to put this all in sort of another larger context of looking at these different flower visiting insects and the different factors that affect their choices. As I mentioned, I'm really fascinated by this concept of floral constancy because for me, the thing that really excites me about animal behavior is how do animals' brains that are so flexible and plastic, and how does learning, in particular in cognition, act as an agent of selection on other species. So the learning ability of bees can actually be a force of selection, of evolution on plant traits. And I find that to be particularly interesting. And bees and flowers are a really wonderful system to get to ask those types of questions. And not only do we expect floral constancy to be influenced by chemistry, but also we know that bees are using scent, they're using flower shape, flower color, electromagnetic fields, a whole range of different floral traits when they're making decisions. And I'm very interested in our research on Kent Island has shown that different bee species are more constant to some plant species than to others. And they vary. So this is Bombus vagans results I showed you earlier, and this is a small solitary bee. And we're looking at that the small solitary bee is more constant to dewberry, whereas the Bombus vagans is more constant to yellow rattle. And what are those different factors that are driving these differences between these species and constancy to particular plants? And what's the consequences of that for the plant in particular? And this all fits within the context on Kent Island. We're developing a pollinator network for the whole island. So we're looking at what are all the plant species that are available, who are all the insects that are foraging there, and what are the connections between those plants and insects. And this was work done by another Bowdoin student, Hannah Scott. She was on Kent Island in 2019, and she sent out all of our summer Kent Island students. They'd spend a couple hours in the middle of every day. They'd pack their lunches and go out all over Kent Island. This is an aerial map here showing you Kent Island. And the different colored dots are different plant species, and the size of those dots are the number of insects that were collected on those different plant species. And we're trying to put together a really comprehensive network for the plants and pollinators on Kent Island so that we can understand, get some insight into what are the different factors, both on the plant side, as well as on the insect side, that are affecting choices that those insects are making, and how does that impact pollination of the plants. And this has commercial implications as well. So Kent Island has quite a bit of lowbush blueberry, and we've been working on, this was a Bowdoin honor student who did her work with blueberry on Kent Island in 2018. And she found that there's a lot of variation between our blueberry patches on Kent Island. So each of these this is sort of the middle of Kent Island, each of these blue spots is a lowbush blueberry patch. And what Rachel found was that different patches varied quite a bit in fruit to bud ratio. So it's sort of a metric of pollination. And we're trying to disentangle, how much of that is due to the insects that we're visiting, how much of it is due to soil quality, or other factors of those particular patches, perhaps just genetic differences between those patches that's driving this variation. And how does where a patch is situated in this network of plants and pollinators influence the rate at which it gets pollinated and the amount of fruit produced by those blueberry patches in particular. And then this is to remind me to say that in this pollination work, we recruit all of our Kent Island summer students, of which we have sort of 8 to 10 every summer. And they spend the sunny hours in the middle of the day when they're not working with seabirds or crabs or whatever else other research that they're doing, learning to identify plants and pollinators all over Kent Island, and contributing to this, what I hope will be a new long-term data set for Kent Island, looking at who our pollinators and plants are and how that network may change over time. So to conclude here, I have lots of students, past and present, who work with me on this research. Collaborators, Anurag Agarwal was my postdoc advisor at Cornell and is a current collaborator on a grant. And then Jack O'Brien's in the math department at Bowdoin. And then I've been funded by a wide range of people, and our work on Kent Island is mostly funded by Bowdoin. So thank you to all of those things. And I'm happy to take any questions. And you may have to shout out because I can't see all of you at once. So feel free to speak up. You may have to unmute yourself to ask me a question.
I have a question for you.
Great.
So I noticed among the species of flowers that you have on Kent Island, as far as my botany goes, there's a difference in the height of the presentation of those flowers, as well as the shape of the flower. So dewberry, as far as I know, is pretty low down.
Yep. It's crawling sort of. Yep. That's how you tell it from raspberries when you're a naive botanist like myself. Yes. So you're interested in how that might be affecting bee preferences?
Right. Their choice. I mean, you know, especially you talked about the 2 different sizes of bees. The smaller one would be able to penetrate deeper, I think.
I think that there. Yeah, I think that there are a bunch of species differences. When we are doing this plant pollinator network, we're looking at, we're measuring a bunch of plant traits as well. Height and density, just numbers of flowers available. You know, there's the species itself has got differences. The dewberry's crawling and raspberry's upright. But then how dense that flower is in that area, we think might also really be important for that bee's decision about what to visit, as well as how many other options there are. So at each point where we collect an insect, we measure I have the students measure within a 10 square meter radius around that particular site where they got the insect, how many flowers are available of that plant species and how many other species are available and flowers of those other species. I have them take a fairly coarse, quick measure, but to get a sense of what are some of those other factors that are probably influencing that choice that the bees are making. And height could certainly be one of them.
I have a question from River Santina, who asks, looking at the soil samples regarding the blueberry patches, have you looked at any mushroom presence differences?
Oh, I would love to do that. I would love to look at soil mycorrhizal impacts. Do you mean mycorrhizae, or do you mean other types of mushrooms?
Both, really. I mean, the mycorrhizae are so interesting to you know, kind of tease out, but anything regarding the mushrooms.
I know so little about fungus, and I am trying to learn more. We've had some students who've worked on fungi on Kent Island in the past. We know that mycorrhizae are really important for blueberry in general. There's a guy at the University of Vermont who does work on high bush blueberry and mycorrhizal effects on blueberry production. We haven't looked into mushrooms that are surrounding blueberry bushes or different soil types much at all so far. We found that our soils are not so different between patches, like they're quite similar in acidity at least, but we haven't broken down a lot of the other factors.
Are there any, I mean, do you see many mushrooms there? Because, I mean, it's not as you do.
We have boletes, and we have, there are quite a few mushrooms. We're there in this, I feel like the best mushrooms are there when I'm there in the fall, which is like not when we're there with students. So I feel like I'm not even getting the best snapshot of when those mushroom diversity, but there's quite a lot of mushrooms there. It's about a 200-acre island, so it's not tiny.
Okay, cool. Thank you.
I have a question for you.
Great.
I know that, for instance, I've done some work with neonicotinoids, and some of the bumblebee species metabolize them incredibly quickly, less than 2 hours. And I was wondering, had you looked at any of these toxins to see how quickly like if they're metabolized as fast as your sugars that they're taking in for flight energy? Do you know anything about that at all?
That's a great question. We haven't looked at metabolic rates of these compounds. What we are doing at the moment, I've got a lab technician who's a microbiome person, and we are doing a bunch of experiments feeding bees these different compounds and looking at how the impacts of it on their microbiota. So that's something that we're particularly interested in, because that could have, so even if they metabolize it quickly, if it's shifting their gut microbiota, that potentially could have even could still have a longer term effect on the bees in terms of their physiology or even their behavior. But that's a great question. And I don't know about differences in the rates at which they metabolize these compounds. But we are trying to start to look into at least differences in how it might be affecting their gut microbiota.
That's very interesting about your research on the gut microbiota, because there's, you're probably a lot more aware of it than I am, but there's quite a following of people who use this term self-medication.
Right.
That that bees are actually visiting these flowers and picking up these secondary plant compounds to protect themselves from microorganisms that are pathogenic.
Yeah, particularly Crithidia. Yeah.
What about mad honey? Have you ever heard of that?
I don't think so. That's honey with something else in it, or it's from a particular plant?
Well, there's some types of bushes like rhododendrons and stuff that are actually have the toxics in them.
Yeah.
You can buy mad honey on the web, and it's sometimes added to drinks to give it alcoholic drinks to give it an extra tang or something like that. I'm not sure it's legal, but like I said, I read about mad honey, and I looked it up on Google and stuff, and there it was. I could buy some if I wanted, but
Honey with uncontrolled plant substances in it, huh?
I'm not sure what well, like I said, this rhododendrons and stuff have some toxins in them normally. And there's places in Europe and Asia that raise those bushes primarily to harvest the honey that's toxic, and then they can sell it at a good price to the fools in the United States. So have you ever done any?
I don't know much about rhododendron and what the toxins that are present in rhododendron are. I mean, anybody who's buying local honey, right, you find that that goldenrod honey in the fall is very different from clover honey in the spring. And if you've had tea tree honey, manuka honey from New Zealand, it is thought to have really strong antifungal, antimicrobial effects due to the compounds present in the tea tree plants themselves. So it certainly does carry over. It wouldn't surprise me at all if there could be pharmacological compounds that have psychiatric effects or on humans that are present in floral nectar. We know there are, I guess I should say, that at high enough concentrations in honey, you could be seeing some kind of interesting effect. It doesn't seem impossible to me.
Well, thank you. Very interesting.
I have a question. Oops.
Wait, I had you before, and now you're muted, Kathy.
There we go.
Now you're on.
Yeah. So I think it was Frank that brought up the question that was a little bit like what I was thinking of, is it's such a complicated thing you've got there with all the genetic differences and the learning that can happen. And so it's a very complicated web that you're trying to tackle. It's really interesting. But I wondered about how long you were in your laboratory experiences. Is there, I heard you mention something about 2-hour exposures. Was it longer than that? Is there an opportunity in those laboratory experiments for the bees to have done any learning?
Well, bees will learn
Good.
in what way? Sorry, you were going to say something else, Kathy.
Well, I don't know what the learning curve is like for bumblebees anyway, I guess. So
It depends on what they're learning. So bees will learn a color or a scent immediately with one, just practically instantaneously. One visit to a flower color and they have learned that color. They're very, very fast learners. In terms of these different compounds, what they're learning, we don't always know. So some of these compounds are distasteful. Cardenolides that occur in milkweed nectar, we think we know to mammals they taste bitter. And we might expect, therefore, that bees could be learning a bitter taste. They could also be learning on a longer time frame, learning, if not feeling well. And that's sort of a different type of learning, right? That would take a much longer time frame than an immediate response of something that tastes bitter. The research that's been done with honeybees and caffeine has shown that honeybees do taste the presence of caffeine at high enough concentrations. But some of these effects at these low concentrations are likely not, a deterrent effect of the flavor, but rather a effect that the caffeine is having on the bees' brains. But that's always a hard thing to disentangle. It takes somebody who's good at neurophysiology to be able to sort of be recording from the antennal neurons of the bees and looking at, or gustatory neurons, taste neurons, and being able to look at, are they able to taste these compounds or not? And then looking at, so they could be, influences on their behavior could be by taste, which could be immediate. It could be a sort of psychological, pharmacological effect, which could be maybe medium term, or it could be, over a long term, they start to sort of feel ill, and that could be a much longer effect. So it's hard to disentangle those things, and we think they'd act on different time frames. Some of the experiments I've talked about are very short time frames, one bee making one foraging bout that maybe is less than 5 minutes. And other ones are, bees been foraging on a solution for 2 hours or even for days in some cases. And then I'm looking at their choices that they're making. And those things could be acting differently on those different timeframes.
Patty, I had a question about your experiments with the pairing, the social interactions.
Yeah.
Well, a lot of bumblebees, they mate on flowers. And have you looked at male-female interactions and how it might be affected by these compounds?
That's a great question. And no, I don't work at all really with the males in my colonies, because they're just kind of a pain in the butt. And all they want to do is mate with females and not make foraging choices for me. So I haven't done that at all. I'd love to be able to do some work with males, because they're, they don't sting. They're nice to be able to do behavioral experiments with, especially for undergrads in the lab, but I just can't get them to be motivated to forage well enough for me to do experiments with them. So everything I've done is just been with females.
Yeah, maybe in the field, it might be easier when males are produced for a given species. And if you go out, especially a lot of the flowering shrubs in late summer, you can find them quite a bit. And maybe you could somehow manipulate a system like that.
And I think that would be, certainly around Brunswick here in the September, all you see are males asleep on everybody's dahlias in the morning and stuff like that. There are a lot of them around. And that would be a great opportunity to get to work with them in the field.
Yeah. Well, we've got some interesting stuff going on.
I had a question.
Great.
So I recently read, I don't have a background in science, but I recently read that like some flowers have this blue halo effect to attract bees, because bees tend to be attracted to the color blue. And so I was wondering if you had to control for colors that bees might like better than others in your experiment.
Yeah, great question. And yes, and we're also thinking about ultraviolet light a lot. So that halo, I think, is that you're thinking of is it's usually when you've taken a picture of a flower with a UV light and a UV lens, that you can see that often flowers that to us just look white and black or something will have these ultraviolet stripes or rings around them that are nectar guides, they're called, for the bees. So the colors that I work with when designing behavioral experiments are ones that have been mapped in bee color vision space. So we've we've measured their reflectance using a spectrophotometer and then looked at where did these colors fall within bee color vision space. So we really have a sense of can it be distinguished between these two colors? You know, do they have any spectra that we're missing? Like, are they ultraviolet reflective or stuff like that? I then also have like lighting in my lab that contains some ultraviolet to it. So because that helps the bees sort of behave more normally in that type of environment, to have some ultraviolet light, more like sunlight. And I have to think a lot about the tops of my arenas have to be, let that ultraviolet pass through and things like that. So the particular colors of the flowers is really important. And it's part of why it's so much easier to work with artificial flowers that are these set color chips that I can order from particular color specialty color companies, because I can control for that. Whereas, you know, even real milkweed flowers that I've grown in a greenhouse are probably varying in ways that I'm not seeing, and might be creating variation in bee behavior that I don't have control over.
Thank you so much.
Can you all hear my dog whining in the background about the ball he lost under the sofa for this whole time? Yeah, sorry about that.
No problem. Lisa Wiley asks if the public can visit Kent Island.
Yes, if you can get yourself to Grand Manan and send me an email when you get there or that you're coming. We'll sort of slot you in with a normal food run or something that we're doing. We still don't know how this pandemic summer, we had to cancel last summer because the border was closed. You have to cross the border. So we still don't know quite what's going to happen this summer. But generally, in more normal times, we're very happy to have people who are interested in coming out to see the field station. If you can get to Grand Manan, which is a lovely place, great whale watching, lovely fishing community to visit in the summer. We're out there the end of May until the end of July, and we can bring you out on our lobster boat, probably fit you in with a food run or something like that to spend the day exploring Kent Island and seeing the place a bit. It's a really wonderful little field station. We house about 25 people, totally off the grid, just solar power. And students do research projects, and we have faculty from the University of Guelph and University of Windsor and Williams College and Kenyon College who bring their students and work out there for the summer too. So it's a great community and place.
Kathy asks, or let me see if it's a question. Yes, okay. People who want to attract native bees are often told to plant swaths of the same species near each other. It sounds like your research in part explains why that is helpful. Do you think it is a question of energy budget of the bees?
That's a great question. I don't know necessarily who, like the decision that's being made when people are advising that per se. I usually, when asked about what to plant, oh my gosh, I have to fetch this ball. I will talk to you while I fetch this ball because I can't handle the whining anymore. Hang on a second. I really encourage people to think about, planting native plants and really importantly, planting ones that range the seasons. So some early flowering spring things into late flowering ones for the males and so forth. So across the whole course of the summer. Because of the way that they specialize, I think that you would be able to increase the efficiency of, we think that the floral constancy, what it does is really means that bees get efficient at extracting nectar or pollen or both from one particular type of plant. And then if they have a large amount of that plant to visit, they can very efficiently forage. And so having both a range of things and then a high density of things obviously is, could be ideal from the bee perspective for maintaining large populations in particular. But I can't speak directly to what